Hello, and welcome to our presentation on our progress on the lab-scale bioreactor design for the production of monoclonal antibodies in a mammalian cell line. Mammalian cells are extremely important in industry because 21 of the top 50 top-selling drugs are produced by mammalian cells, and CHO cells produce 60 to 70% of those products. It's important to produce some bioproducts in mammalian cell lines because the post-translational modifications that mammalian cells can perform are necessary for the product to work in the human body. E. coli and yeast cells just aren't efficient enough at doing post-translational modifications that allow proteins to be recognized by our complex human body. And so some industry challenges that are currently going on with biosimilar production is that it's hard to produce a pure product, and there are currently a lot of advances in chromatography in order to do that. And of course, producing at 10 grams per liter is a huge jump in product concentration in our bioreactors, but there's always more work to be done. So in our lab, we are focusing on 3 different areas. Sherrie Riser is focusing on how to grow our cells and produce our product within a suspension cell culture. Alec, Wallisch, and I are attempting to transfect our product into mammalian cells. That way we can grow it in Sherrie's bioreactor that she's developing. And Laura Weller is currently working on hydrogel research in order to deliver our product to our patients.
We chose to look into 2 different avenues of optimizing cell growth. We chose to look into suspension cultures and chemically defined media. We looked into suspension cultures because they offer higher cell densities and viabilities than traditional adherent flasks because they are not limited by the surface area of the flask. So in the image on the right, you can see our suspension culture setup. We have non-adherent flasks sitting on a rocker that continuously rotates to provide agitation to the media. We also chose to look into chemically defined media because our current media contains fetal bovine serum, which is very useful for growth nutrients and growth factors, but it can make the downstream processing of your product very difficult because it contains extra proteins and enzymes that have to also be separated. So after some research, we chose to use the additives shown on the slide for our chemically defined media because they've been shown to encourage cell growth and to inhibit apoptosis effects in the cells. So we chose to do an adaptation study for our cells, starting with a 75% serum-containing media and a 25% serum-free media. And the plan was to decrease the concentration of the serum-containing media from 50-50 to 25-75 and then to 100% fully serum-free media. But after about 6 days, we noticed a significant clumping problem with our cells. And a representative image of that is shown on the right. So in the suspension cultures, the cells have a tendency to aggregate into balls, which makes it very difficult to obtain accurate cell concentration data through counting the cells. So the adaptation study has been placed on hold while we formulate anti-clumping agent concentrations in our media in order to remedy the clumping issue so that we're able to collect accurate data.
Plasmids stand as a bottleneck in our progress. They're expensive, and to buy from a company, they cost about $70 per trial, which is a little bit more than we can afford for all of the things that we want to do. So we've had to seek a more cost-effective alternative for our plasmids. We've done this by taking E. coli and growing GFP plasmid inside of them and purifying it with a plasmid isolation kit from Thermo Fisher. Doing this, we've gotten the price down to about $7 per trial, which is great progress, but we're looking to do more. We're trying to get the price to about 30 cents per trial, so that way we can do all of the experiments we want to without having to worry about cost as much. The only issues that come with this is the purity can be a problem, as well as the plasmid concentration. If the concentration is too low, we can't actually get enough DNA in with our cells when we electroporate them. And if the purity isn't there, it doesn't really matter because the DNA in the plasmid doesn't necessarily do what we want it to do. So we've had to work and troubleshoot our efforts to overcome these problems.
After collecting our plasmid from the E. coli cells, we need to transfect it into our mammalian cells. We do that by electroporation. After electroporating our cells, we grew them a 24-well plate and took data 2 days later on their fluorescence. The results you can see here are that our most useful voltage for transfecting cells was 280 volts. This was the factory recommended setting, and we tested some voltages higher and lower just to make sure that was correct. And so what you can see here on the graph in the top left is our control fluorescence and our 272 voltage fluorescence. And so what's key here is the graph on the bottom left. You can see that the control has no fluorescence above 10 to the fourth. And you can see that the 272 volt sample does have plenty of cells that are fluorescing much above that. And so our data here shows that the 272 volts can fluoresce GFP more than the non-transfected cells naturally. And so our key variable was how much more of them could fluoresce compared to how much of them in the control were. And so here on this next graph, you can see that with the control having, of course, 0 fluorescence above normal fluorescence, all of the other cells did have fluorescence that occurred above the maximum control fluorescence. And so our 272 volt experiment produced the most number of cells as a percent of the sample that were above the control's ability to fluoresce. And so by looking at this, we determined that future transfections should continue to be performed at the factory recommended voltage.
Current monoclonal antibody therapeutics require intravenous delivery and have difficulty targeting specific areas. To further this research, the 3D cell culture is needed. Current lab techniques allow for growth of cells in a flat monolayer in a flask or in suspension, neither of which are representative of a 3D tissue culture. In order to create a 3D tissue culture, I used a collagen-1 hydrogel to provide the 3D structure necessary to grow the CHO cells. I created the collagen hydrogels in order to optimize the viability of the cells. To do this, I varied collagen concentration and cell plating location. To measure the viability, half of the sample were added to rubidium iodide and then measured under flow cytometry. This allowed the cells with a breached cell wall, indicated by rubidium iodide, to be measured. This is the graph of the results from the flow cytometry of the collagen hydrogels. As you can see here, from columns A, B, and C, the highest collagen hydrogels had the highest viability, while the lowest concentration did not have the lowest viability as expected. As then looking at columns B, D, and E, the highest viability came from when the cells were plated on top as well as inside of the hydrogel. It was expected that the cells plated on top of the hydrogel sample would have the highest viability, but instead it had the lowest. These results may be due to low recovery of the hydrogel cells before testing. Future work of our growth research will be optimization of the serum-free growth media and anti-clumping agents. Future work of our product research will be to generate stock supplies of the GFP plasmid and to transfect the suspension cells that have been adapted to the serum-free growth media. For the future work of the product delivery will be to characterize the hydrogel material properties. Thank you. Are there any questions?